We bring the lights. Lights down? Yeah. Thank you. All right. Last before lunch. My name is Brad Seibel. I'm an animal physiologist, and I'm going to talk to you about the effects on animals of ocean deoxygenation in combination with warming. This picture is actually a much prettier picture than it appears here. This was taken by Stephanie Gordon, who gave a talk here last year. She's from Open Boat Films, an outreach filmmaker. But it's a little blown out here. So let's see. So three short parts to the talk. The first is on the physiology. Animals have specific evolved oxygen transport capacities that contribute to their total aerobic capacity as well as to their ability to tolerate low oxygen. And I'm going to describe a database that I've spent far too much time over the last year putting together of every instance of hypoxia tolerance I can find in the literature for every animal on the planet, well, in the water anyway. The second part is on biogeography. How do we relate hypoxia tolerance and that aerobic capacity to the distributions of organisms, both latitudinally and vertically? And the last part, I want to talk about a few specific claims that have been made regarding ocean deoxygenation, including does carbon dioxide influence metabolic scope? That's something that's tossed around in the literature without much evidence. Are fish going to shrink? That's actually been tossed around a lot. Due to increased warming, it's projected that fish will shrink. And lastly, that polar regions relative to lower latitudes have excess oxygen. And I imagine I will be able to debunk all of those, at least in part. So this has become an issue. Ocean deoxygenation has become an issue, a hot button issue in the last several years. The idea is that as the ocean warms, the solubility of oxygen decreases, and that contributes to expansion of oxygen minimum zones. So this is a transect from Alaska down to the equator. Going from Alaska down to the equator here, you can see that much of the eastern Pacific Ocean is extremely low in oxygen. At the eastern tropical, everything within this purple region is 25 micromolar or less, which is about 0.8 milligrams per liter or 2 kilopascals partial pressure at 10 degrees. Units have been the reason that creating that database has taken the better part of my life. I've gotten very good at converting them, but it's still not trivial to think about different units, so I'll try to help you along with that where I can. 2 milligrams per liter is often touted as hypoxia without much justification for it. There's a lot of oxygen concentration units that are, or definitions of hypoxia that don't have a lot of biological meaning, so I'll try to provide meaning as we go along. Closer to home, we've got the dead zone that occurs here, especially seasonally in the Gulf of Mexico. Everything within this black line is 2 milligrams per liter, which is 7 kilopascals at 30 degrees. So the entire eastern Pacific Ocean is lower in oxygen than this dead zone for the most part. There are parts in there that reach anoxia, but for the most part, the dead zone is not actually a dead zone, and depending on which animals were exposed to it, it would have no or extreme consequences. But the dead zone here in the Gulf of Mexico does create fish kills and is something that a lot of people are very interested in locally. So hypoxia tolerance, or just tolerance of oxygen in general, is entirely a matter of a gradient. So at sea level, you've got 21 kilopascals of oxygen partial pressure, and as you move from the air that you breathe in down to the alveoli in the lungs, into the blood, to the venous blood after it's gone through the tissues, you lose that oxygen partial pressure. You have to have a gradient from the air you breathe in down to the mitochondria that are using the oxygen in cellular respiration. Humans can tolerate about 10 kilopascals as the lowest partial pressure that we can tolerate. That's the equivalent of 5,800 meters of altitude. Above that, most people require excess oxygen. They need to bring oxygen with them. So No, that is not me. I am not so crazy as that. So 10 kilopascals, that's higher oxygen than you find in the entire Eastern Pacific, as I just pointed out. So animals are much better at this. And in fact, 2 kilopascals is that 25 micromolar purple region I showed in the oxyminimum zone. So animals living in the oxyminimum zone manage with only 2 kilopascals to get oxygen all the way to their mitochondria, where the oxygen partial pressure is, in humans, about 1 kilopascal. So somehow they manage with that smaller gradient. And the way to do that is fairly well studied in organisms. If you put an animal in a chamber, here's that same squid from the original picture I showed you, in a small chamber on board a ship in a water bath there, and we're measuring the amount of oxygen in the chamber over time. And as the oxygen in that chamber declines, most animals can maintain a fairly constant rate of oxygen consumption. And they do that as the oxygen declines by increasing blood flow, increasing water across the gills, increasing the heart rate, adjustments of acid-base physiology, etc., and lesser well-known adjustments in the mitochondria themselves. And you can do all that down to some critical level where you can no longer meet that routine rate of oxygen consumption. And at that point you fall off and your rate of oxygen consumption declines with declining oxygen. If you increase your activity level, that rate is going to be higher, and your critical oxygen partial pressure is also going to be higher. You're less tolerant of low oxygen if you're running fast or swimming fast. And if you increase the temperature, same thing. The rate of metabolism goes up and the critical oxygen partial pressure goes up. Deoxygenation is pushing the environmental oxygen availability to levels potentially below that critical oxygen partial pressure. So I've created this database. It's currently got more than 600 measurements from about 325 species. So many of those are measured at multiple temperatures. That's why more measurements. So they span the range from Antarctic animals at -1 degree up to tropical animals at 32 degrees Celsius, across the entire size range of metazoans, so copepods up to sharks and large squids. Animals from all major ocean basins, from the surface to the deepest depths, every major habitat type from intertidal zones to hydrothermal vents to oxyminimum zones, and a few here in the Gulf of Mexico, and every major phylogenetic group. So that's going to be the basis of this first physiology section here. So temperature describes most of the intraspecific variation in that critical oxygen level. So this is on a log scale, the critical oxygen level as a function of temperature. Each of these lines is a different species. So you can see that the critical oxygen partial pressure is species specific. So it doesn't matter what temperature you're at. There are tropical animals that have the same critical oxygen level as polar animals. And this star represents where humans would fall on that map at our body temperature of about 37 degrees. So we are not especially hypoxia tolerant, but not the worst. Once you normalize to a common temperature using those relationships, you can see that ocean basins are very different. So animals from the Eastern Tropical Pacific oxygen minimum zone don't overlap almost at all with the rest of the crustaceans that have been measured. So these are just crustaceans, all normalized to a common temperature. The Antarctic animals are not on this plot because if you normalize them to temperature, they're off the map. They have a critical oxygen partial pressure greater than air saturation, which means that at 15 degrees they would not be with us any longer. But chances are something else would kill them before oxygen at 15 degrees. So, but I'll come back to that. So these animals are not normalized to temperature because they live in the lower oxycline. So in an oxyminimum zone, the oxygen declines with depth to about 200 or 300 meters, and then it stays low, and then around 700 or 800 meters it starts to creep back up again. And these guys live at that lower oxycline. So when they move shallower, the water gets warmer and the oxygen gets lower, which is the inverse of what most animals experience. Warmer water, typically their critical oxygen partial pressure increases. These guys all decrease. They're actually more hypoxia tolerant in warmer water. So there's specific adaptation to their specific environment. The other point to take away from this slide is that hypoxia tolerance across species is not dependent on the oxygen consumption rate. You can have very fast swimming animals that are just as hypoxia tolerant as some of the most sluggish animals in the world. All right, let's see if this plays. This is not critical, but there it is. So this is one of those shrimp in the lower oxycline. This is Ganates. It's a reasonably active midwater shrimp, and it lives at about 800 meters. So if it moves a little bit shallower, the oxygen actually declines. So this is oxygen in micromolar, and we're talking about very low oxygen levels here. So from 8 to about 4 micromolar as it moves up in the water column. So it gets warmer, but it also gets lower in oxygen. Their metabolic rate is lower. Their metabolic rate is lower at colder temperatures, as you would expect, but their critical oxygen partial pressure is lower at warmer temperatures. So a very specific adaptation. So while all the other lines are going up, theirs goes down with increasing temperature. Body size does not have an effect on hypoxia tolerance. We've measured everything, as I mentioned, from copepods to that shrimp that I just showed you, larger squids, and my student Alyssa Andres, is she here? Nope, she's flying. She was giving a talk on some of this work up at a fisheries conference. So anyway, this is her chamber that she set up down at Mote Marine Laboratory with a juvenile black-tip shark. So the point here is large animals are not less hypoxia tolerant, as some people have claimed, and this comes back to this idea of shrinking fish. So remember that in a moment. Lastly, phylogeny does not seem to influence hypoxia tolerance either. There are some claims that fish are less tolerant than lower, quote-unquote, lower organisms. It doesn't seem to be the case. There are representatives of every major phyla that are extremely hypoxia tolerant, if that's what they're exposed to, and there are some that are not, if that's what they're exposed to. All right, so part two, how do we relate this hypoxia tolerance to the biogeography of species? This is a description of a metabolic index that I published with Curtis Deutsch as lead author. He was one of our speakers here a year or two ago now. And the idea is that the ratio of oxygen supply to demand is this metabolic index, and you can calculate that. So here's oxygen supply. This describes the temperature effect on metabolism. This describes the body mass effect. And this A naught includes the physiology that I've been talking about. So whether species have higher gill surface area and better blood oxygen binding capabilities, that's normalized in this coefficient here. And you can calculate the E naught and A naught if you know the critical oxygen partial pressure, because by definition, at the critical oxygen partial pressure, oxygen supply exactly equals oxygen demand. So this metabolic index we interpret as a potential factorial aerobic scope, meaning the potential factor by which an organism can increase its metabolism above its resting needs. So this is that critical oxygen pressure measurement I showed you before, where they maintain a constant rate down to some critical level and it falls off. The increase in that critical level with temperature. So these black numbers are critical oxygen partial pressures at those different temperatures. The metabolic index is just the ratio of the partial pressure at air saturation relative to, or whatever oxygen habitat they're in, to that critical oxygen level. So in air saturated water, the metabolic index at 30 degrees is 3. At 24 degrees it would be 3.5, etc. So this is actual data for black sea bass that I'll come back to in a moment. Another way to look at this, so here's that black sea bass data, critical oxygen partial pressure increasing with temperature. The metabolic index is the ratio of air saturation to that critical oxygen level. And you can see that it's species specific. So this is Atlantic cod. At its upper temperature range, it has a similar metabolic index. At a lower temperature, it has a higher metabolic index, a greater potential aerobic scope. And a tropical fish here, at its upper temperature, has a similar metabolic index as well. Here's that 2 milligrams per liter line that a lot of people like to cite as hypoxia. These fish would, at lower temperatures, just be able to meet their routine demand at 2 milligrams. In other words, if they were in 2 milligrams, their metabolic index would be 1. Whereas this tropical fish would actually be able to tolerate 2 milligrams relatively easily. And that's because 2 milligrams is a moving target. Oxygen concentration changes with temperature. This is a deep sea eel pout living in an oxygen minimum zone off California. And even in extremely hypoxic water, it has a metabolic index of 4. So it would have a metabolic index of about 20 in air-saturated water, but it doesn't live in air-saturated water. It lives at depth in low oxygen. So oxygen concentration is not a particularly useful measure of hypoxia tolerance. Animals respond to partial pressure. Air-saturated water at 30 degrees holds 6.4 milligrams, but that would only be about 13 kilopascals at lower temperatures. So, all right. So just to drive that point home, there's some literature that suggests sharks in the Gulf of Mexico here would avoid water with oxygen concentrations less than 3 milligrams. But this data that Alyssa has collected shows that at about 20 degrees, they could just barely tolerate. They could maintain their resting rate of metabolism. But at warmer temperatures, they would require much higher oxygen than 3 milligrams. All right. So for a handful of species, there are measurements of both aerobic scope, which you measure by putting them in a flume, basically a treadmill for fish. You put them in a flume and turn the current up and make them swim against it until they reach a maximum metabolic rate. So that's maximum metabolic rate going up with temperature. And this is their resting metabolic rate going up with temperature. And the difference between those two is aerobic scope. For a handful, we've measured both aerobic scope and the critical oxygen partial pressure. So we can compare actual aerobic scope to this estimated aerobic scope with the metabolic index. And what you see is that the two completely independent measures of factorial aerobic scope are very similar. And this tells you that the two are mechanistically connected. So the same gill surface area and some of the blood adaptations that are required to make you hypoxia tolerant are the same ones that allow you to increase your metabolic rate during activity. It also tells you that at any temperature, animals are utilizing their full factorial aerobic scope, and that any decrease in oxygen, so any hypoxia, would potentially limit that aerobic scope, and any increase in temperature is going to decrease that aerobic scope as well. So you're going to have a reduction in potential for growth, reproduction, and activity with hypoxia or warming. And they increase towards some critical value. And this critical value we call the critical metabolic index is species specific as well, but it's the ratio of energy needed for sustained activity out in the wild relative to rest. In other words, if you have a population of animals, the lowest metabolic index that population experiences is their critical metabolic index. The operational definition is the metabolic index above which 90% of individuals of a given species have been reported. So to show you what that means, this is data for the Atlantic cod again. So all the blue dots on here are the distribution, the known distribution of the species. There's a western population and an eastern population, and their southern limit is bound by a critical metabolic index of about 3. Interestingly, the two populations behave differently over seasons. So in the western population, they migrate between about 41 degrees north to 37 degrees north in the spring, from fall to spring, so along this line. And this black line is that metabolic index of 3. So they follow that line seasonally. Latitudinally in the western population, and over depth in the eastern population. They migrate between the surface in spring to about 200 meters in the summer or fall, again following that metabolic index of 3 line. So it seems to be an important range limit for this species. Similarly, we can identify critical metabolic indices for any species for which there's sufficient distribution data available. For a tunicate, we've identified about 1.5 that seems to bind it or restrict its range limit. For the spotted cat shark, it's about 2. For the pink shrimp, about 3. My student, Alex Burns, is she here? Oh, she's teaching. All right. 0 for 2. She is starting to make measurements of both aerobic scope and critical oxygen level for a variety of shrimp here in the Gulf of Mexico. So we hope to be able to contribute that to some of the modeling efforts that are going on around here. Anyway, so the critical metabolic index for a variety of species, we've identified about 30 species that have the physiology data as well as the distribution data necessary. You get a range with peaks between about 2 and 4 with an upper tail, a critical metabolic index of 6 or 7. And it pretty closely matches the sustained metabolic scope measured by Killen et al. For a variety of fish species. So we think that it is an important range description for these species. This is one slide to explain a very large and complicated project, but this is work that I did with Chris Roman, who has this instrument he calls a wire flyer. It's basically a towed CTD array. It goes up and down in the water column and makes pretty maps of oxygen and temperature. And Karen Wishner, who had a multiple opening closing net system. And we did horizontal transects over about 40 kilometers. Those are seen here. The colored lines are where the wire flyer was towed, and these black lines are where we